Good morning. Thank you for giving me the opportunity of presenting our work here. My name is Wang. I'm a PhD student from University of Maryland and graduating this year. And today I'm going to talk about mechanical guidance through cell-cell and cell-surface contact during collective cell migration. And this is a collaborative project between Dr. Wang's lab at UMD and Dr. Kara's lab at National Cancer Institute. So I would like to start with why we are interested with collective cell migration. Cells collectively migrate to heal wounds, to develop tissues and organisms, and also cancer cells may use collective migration to invade neighboring tissues. And so collective cell migration is a ubiquitous phenomenon. And here are two examples on collective cell migration. The left one is actually from my colleague Rachel Lee. She works on migration of epithelial cells. And those cells form a sheet and move together towards the left side of this figure. And the second case is actually an in vivo experiment. Shows that tumor cells can actually move out from the tumor and invade neighboring tissues. In both of these two cases, what we found is that they form this strand-like structures, which we call multicellular streams. And our aim is to understand the cell-cell interaction when they are migrating inside of such multicellular streams, and also to understand how the external substrates affect their migration. And to study that, we use our model system, which is dictyosteroid beta cells, which are those bright dots in this movie. Usually those cells are single amoeba cells moved individually. And upon starvation, they will start to collectively migrate to form streams and migrate towards the aggregation center. Eventually, they will form a multicellular structure that contains hundreds of thousands of cells in it. And if we look into the streams that they formed, and this is streams that cells labeled with live at, and we will find that cells actually form head-to-tail alignment in the streams, in which case that the leading edge of one cell is directly contact with the tail of the other cell. The mechanism that they form such a head-to-tail alignment has been previously studied, and there is a chemical signal relay pathway that's been discovered. In one word, that all the cells in the field can sense the chemoattractant gradient and move towards the chemoattractant source. And also, at the same time, the cell can secrete that chemoattractant, and so that sensed by the neighboring cells, and they move towards the tail of that cell. So this is how the cell initiates the streaming process and recruiting new cells into the streams. While after the cell entering the streams, they actually form cell-cell adhesion and also cell-substrate adhesion. And the first question that we would want to address is, in terms of the cell-cell interaction, how the actin dynamics and the shape dynamics of this cell coupled through the cell-cell contact. To understand cell-cell coupling, we first want to understand how individual cell would move. And my previous colleague, Megan Driscoll, developed a tool to extract the shapes of cells from a movie and do measurements along the cell boundary. And when we overlay the cell boundary, we will see that the protrusion extended by one cell initiates at its front and can travel to the back of that cell and forming those small small traveling of those bumps on the cell boundary. So we extended this analysis to analyze the streaming cells, where the cells actually move inside the streams. And we extracted the boundary of two directly contact cells. And what we see from the overlay of the cell boundaries, that the protrusions generated by the leading cell front can travel across the cell-cell contact region all the way to the back of the follicular cell. And to better quantify this process, we calculate the curvature along the cell boundary, where the represented in color, where the red color shows the bumps on the cell boundary. And we plotted the change with boundary position, where we can find two high boundary curvature areas, which are the front and the back of the cell group respectively. And also there are some local bumps, which are the protrusions that we want to study. So this line is from one single frame, and we add in the next frame and the next, and continue to the whole entire movie. And what we saw is the protrusions that generated from the front of the leading cell can travel all the way to the back of the follicular cell. And we wonder what's the underlying driving force for this cell-cell coupling. And our hypothesis is it's related with different actin polymerization cells. We use similar analysis method to extract the boundary and calculate the actin intensity along the cell boundary. And we obtain these two chemographs. I won't go into details of them. I just want to point out that for individual migrating cell, they have polymerized actin at their front, but also we see F-actin on the side of the cell. While after the cell entering the stream, the streaming cell may mostly just polymerize their actin at this front, where they actually form direct contact with the other cell. And so we see the cell-cell coupling through cell-cell contact. And we also know that cells form direct contact with the surface. So the second question we want to address is how important is the cell surface contact to the cell-cell coupling? And we started with the simplest case, which is a pair of cells that form direct contact. And we compared adherent pair of cells adhere and migrate on glass, and for the pairs of cells that migrate on pad-coated slides. And as I mentioned earlier, the adherent cell shows a wave coupling, and the wave transmits propagates through one cell to the other cell along the same direction. While on the suspended cells, they initiate protrusions at the two free ends, and then the protrusions travel along different directions to the cell-cell contact region. And we also looked at the actin polymerization in those cases, and we found that adherent cells may have F-actin at the cell-cell contact region, while suspended cells have more F-actin at the two free ends. So to simplify our finding, we found that with cell surface adhesion, cells are aligned in a head-to-tail alignment, and then they generate protrusion, and that protrusion can propagate from one cell to the other cell, and along the same direction. And while for suspended cell, they generate protrusions at different ends, and then the protrusions travel along different directions. And these two alignments, we wonder how they change the overall migration pattern of the Dicty cells. So are the comparison between the adherent cells and suspended cells. And we can see that the suspended cells, they don't, instead of forming streams, they form clumps. And they migrate, they aggregate by merging smaller clumps into larger clumps. And if we look at the F-actin distribution on the clumps, and we find that they have most F-actin at the periphery of the smaller clumps. So to summarize what I have to show you today, firstly, we find that cells can coordinate their motion by coupling the waves, coupling their shape dynamics. And that coupling depends on proper cell surface adhesion. And secondly, we find that intracellular actin polymerization actively responds to the change of cell-cell contact or cell surface contact. So with that, I would like to thank you for your attention, and also thanks for the whole LOSA lab and parent lab, and also our funding sources. Thank you. I will take questions.
Very nice. With regard to the gradient sensing itself, is there a front-to-back sensing along the line, information transfer, or is there a leader effect? And then how is the gradient information transferred to the actin motion?
So it's the front-back gradient difference that's sensed by the cell, and it can sense very shallow gradients, the chemoattractant gradients. And basically it has a receptor on the cell boundary, and which can sense and then zoom out and amplify that signal, and then it can tell which way to go.
And is it known how that information is coupled to the collecting actin motion?
You mean how it affects the actin polymerization? I'm not quite sure if it's very well known.
Hi, this is really interesting because have you studied many other surfaces? Because a lot of biology is done growing cells on agar, soft or solid.
Right.
So I think there's going to be a lot of trouble comparing results if one group does it on different surfaces and in the other group.
Right. So actually we have studied a lot of different surfaces, and I just gave two extreme examples, like on glass and totally no adhesion. And we also studied Teflon-coated surface, BSA-coated and polylysing surface. And what we found is the adhesion area on those surfaces are actually similar. And the speed of cells on those surfaces are also similar. The only thing we found is how fast they form that the aggregation pattern. So on some surfaces they tend to form smaller aggregates and then merge into larger ones. And for some surfaces like polylysing, the aggregates a little bit slower. But the whole, it's just the dynamics of the pattern changed. But the speed and other things are the same. And we also studied cells migrate under agar gel. And we found they fully attach to the gel, of course. And also we found the similar wave transmission through cell when they are moving under agar.
Let's thank the speaker again.